Good morning, good afternoon, and good evening, everyone, and welcome to our webinar titled A Comprehensive How-To Demonstration of Higher Throughput Excitation-Contraction Coupling Investigations. This webinar has been sponsored by Ion Optics, so a big thank you to them for helping to make this event possible. Joining us today, we're fortunate to have Dr. Michiel Helmes, the co-CEO of Ion Optics, and Dr. Diederik Kuster, an assistant professor in the Department of Physiology at the VU Medical Center in Amsterdam. They will share best practices and new techniques in high-content, higher throughput investigations of excitation-contraction coupling in isolated cardiomyocytes. I'm Sarah McFarlane from the events team here at Inside Scientific, and I'm very pleased to be your host for today's event. Now, before we get started, I would like to share just a few housekeeping notes to get you the most out of the webinar today. First, this webinar is being recorded, and resources will be made available following the event. Next up, if the webinar panels look too big or too small, you can zoom in or out on your internet browser to adjust the viewing area. You can also resize some of these panels or make the media panel full screen. Please send questions, thoughts, and comments to us via the Ask a Question box next to the media window at any time. You can also take a look at the resources panel, where you'll find a few links and suggested readings associated with today's event. We will also be running a number of audience polls during the webinar and a survey at the end, so please chime in and share your perspectives with us. And finally, if you do happen to experience any technical issues during the event, the easy fix tends to be a simple refresh of the browser page. This should successfully reestablish your streaming connection so you can see and hear us clearly. However, if this doesn't work and you continue to have issues, please use the Ask a Question box to communicate your issue with our team, and we'll help to get you back up and running. Before we move on to our presentation today, we are going to run a quick audience poll. So which of the following animal models do you most often use? And here the options are mouse, rat, human, other vertebrate, and invertebrate. So I'll give everyone a couple seconds to answer that poll. Okay, great. Thanks so much for participating, everyone. And now I'm pleased to introduce Dr. Michiel Helmes. Michiel, the floor is yours whenever you're ready.
Hello, everybody, and welcome to this webinar. The webinar will consist of two parts. I will do the first part where I show you actually how to collect data with the system and how to best do that. And then Diederik Kuster from the University of Amsterdam University Medical Center will take over and show actual cool data that we collected with the system. I want to show you basically how in a few hours time you can get to this graph. The webinar will be, my part at least, will be built up with a short introduction about isolated cardiac myocytes as a research model and why it's important to increase our throughput. It will hardly be necessary because otherwise you would not be watching this webinar. And then I'll go through the timeline of a typical full experiment, isolation, the acquisition, the data analysis, and some simple data export and plotting. And I'll show you some examples of published results. Why do you want to look at cardiac myocytes? It's the smallest functional unit of the heart, and it still functions after you isolate it. It's been around for a long time, this preparation. This picture is from 1976, and there was the first publication I could find that showed calcium-tolerant isolated cardiac myocytes. You give them an electrical stimulus, there will be an action potential, it will release calcium, it will contract, it will take up the calcium and relax again. So it works just like the heart. I got this reference from Brady in Physiological Reviews. It's a really old article. It's actually still a pretty good read. Why do we want to look at calcium contractility? Calcium contractility, because it allows us to look at EC coupling. I took this picture from a review by David Eisler et al. And it shows you a number of the well-known elements, the SR, phospholamb, and sodium calcium exchanger. And we know so much about the effect of these things on the actual shape of the contractility and calcium transients that by looking at those transients and the shape and the kinetics, we can tell, especially with some perturbations, it tells a lot about how the cell is actually functioning, or how its function has changed in disease, or whether you want to see the effect of a compound. The technique is well established. I mean, the first isolation was in '76. People got pretty good measurements by the end of the '90s, and it's only been getting better from there. So it's well established, and I think we can agree that it's a great assay to study the effect of drug disease or mutations on, for example, the sarcomeric proteins. The downside, which has made it a bit unfashionable at times, is the quality of the cell isolation has a strong effect on the results. If you have good isolation, you just get somewhat different data than when you get a bad isolation. And that's resulted in labs sometimes producing contradictory results. But then again, that's nothing new to science. The other downside is the number of cells you could do. The throughput with the old systems was just too low to detect subtle differences, and you couldn't use it really to screen compounds. So pharma companies had to move to different cell types to actually being able to study cardiac receptors. We hope that at some point we can change this so that the cardiac myocyte gets the thing to test drugs on. As I said before, the conventional methods are time-consuming, so you don't get enough data. But there's also a conflict in everybody is pressuring you to reduce the number of animals that you use. On the other hand, when you send your data to a reviewer, they want to see a high enough N number so you can substantiate your claims. You can solve this by just collecting many more cells from the same animal. If you increase the throughput, you can obviously screen more compounds, or you can even do a combination of treatments, and you can use it, for example, to refine, trying to figure out a signaling pathway. It allows you to advanced statistics, so beyond your regular t-test or paired t-test, you can look at the distribution of population, whether it's Gaussian, binomial, maybe skewed, or you can do principal component analysis to look for clusters. There's one article I'd like to recommend in particular, and that is the article of Sickle et al., because it gives you a method where you don't have to take the number of animals as your N, but it doesn't go as far as every cell is part of your N, but it looks at how much the data cluster per cell isolation, and it gives you the appropriate number to use for your statistical analysis. I highly recommend this. I think it's in the resources provided. The only way to really increase throughput, unless you want to put 10 systems next to each other and have 10 analysts working on it, is to automate it. You automate it by having the appropriate hardware and have software to drive the hardware. That takes over a lot of the work. It improves throughput because computers don't get tired, and you don't have to switch between the eyepieces of the microscope and the XY stage to your screen. The computer can take most of the work out of your hands. Another important thing it does, it takes out experimenter bias. Everybody knows if you have a different person, they'll pick different cells, and that creates a bias. If you have poor isolation, but you really need your data, you tend to be a bit more flexible in which cells you include and which cells you exclude. A computer doesn't have those problems. It just follows the rules that you set, which thresholds you set, and what boundary conditions you set. If you increase the throughput of your data, you also need to improve your analysis, because otherwise that becomes the next bottleneck. If you automate the analysis, it has one great advantage. You get rapidly improved feedback on your project, which allows you then to correct the project at the right moment in time. I think we've all been in that situation where you did weeks or months of work and then you found out you should have done something different from the get-go. If you get rapid feedback on your data, it allows you to correct it at the onset of your project. What do we do the acquisition with? We use the Cytocipher microscope. Cytocipher was actually a spin-out from Ion Optics with the sole purpose of developing the system. It works now, we're selling it, and we actually spun it back in. But we like the name and the logo, so we kept it on the microscope. We do the same reliable high content measurements that you use from your Ion Optics systems. You measure sarcomere length at 250 times per second. You can measure ratiometric calcium at 250 times per second. We process the images in real time, so you get a good old WYSIWYG. What you see is what you get. When the experiment is done, you know that your data are good. To really increase throughput, we decided to move the objective and not the sample. If you move the sample, the solution starts to move, so you have to do it slowly, carefully. If you move the objective, you can do it fast. It takes us actually only 50 milliseconds to get to the next field of view. Putting it all in a box allows you to make it light tight and temperature controlled, so you can turn on the lights in the room if you do your experiments, and you don't have to perfuse anymore to keep your temperature constant. It also allows you to have a much more consistent temperature, which reduces the variation in your data. We also have a mode where you can do fully automated cell finding, where you click the button, you walk away, you come back 10 minutes later, and 30 or 40 cells will have been measured, and it will give you over 80% of good measurements, so it works well. It's still a general fluorescent system. You're not stuck with just doing Fura2. You can use any other regular dye as well. And that allows you to do experiments like these, where we double loaded the cell with a folded sensitive dye, fluofold, and a calcium sensitive dye, ROD2, where we also measured sarcomere length. And you get your action potential, your calcium transient, and your cell shortening. So what am I actually going to show you? The idea of a project in the day actually came from a student, Jerome Fourier, that Johnny Perez, one of my colleagues, met in Iceland at a conference. And he started to tell very enthusiastically about how he used the system. So yeah, this idea, I wanted to put it in my presentation, but I didn't have a lot of time. So just in 2 days, I designed the experiment, made the solutions, and did the isolation. And by Thursday, I'd analyzed it, I graphed it, and I wrote it up, traveled on Friday, and he presented it on Saturday. So here I'll walk you through the timeline of a typical day. We start at 9 o'clock, and we do the isolation of the cells. That's usually done by about 11 o'clock, but then we still have to plate the cells. We do our experiments in 35 millimeter dishes, and we leave them in the incubator for about an hour so that they get nice and stable for the rest of the day. So after lunch, the cells tend to be ready for loading, and I was being kind of cavalier about it. I mean, we do calcium measurements so often, of course I could do it without any special preparations. Lo and behold, you don't take it seriously, it goes wrong. The first dish, we had poor loading. I had to do another dish, it's still not great. And I actually collected cells with from this dish, 25 cells before and after ISO, and the contractility data were great, but the calcium data were kind of garbage. I continue today with this 3 and 4, the cells started to deteriorate, and I was a bit despondent, but I thought I only had 1 day to collect my data for this webinar. Luckily, there was another isolation, and we took it a bit more seriously this time. You can't take Fura loading for granted. So we aliquoted everything again. We tried to use different dyes, and dish 1 was still poor. With dish 2, we made progress, and with dish 3, we finally had a decent loading. We actually used Fura4, which is slightly less sensitive to calcium than Fura2, because we wanted to do an ISO experiment where the peak calcium gets pretty high, and Fura2 is better at measuring that. Dish 4, we had another good loading. We used a different dye, Fura2 long retention. We want to practice with this, because for a compound screening, we want to be able for a very long time in the multiple dish. And between 4 and 4:30, we did our data analysis, and we graphed results. And now I will show you examples of all these parts of the process. This is a movie of how you do the mouse heart isolation. It was made by Adam Potato, my colleague who also works in Tim Dollmeyer's lab at the University of Missouri. And he's good at mouse isolations, but mouse isolations, no matter how you twist and turn it, they're still hard. It requires technical skills to cannulate the heart. You need to be careful about it. And this webinar, this movie, by the way, is available via our website, and I think via the resources. We also made a webinar before where different experts in cardiac myocyte isolation talk about how to isolate these myocytes. Most people can learn this, and then the hard part is still can I digest it right. Things have gotten much better with the advent of Liberase, because it allows people to do more consistent measurements. But you still have to pick the digestion time right. If you under-digest it, you will have low yield, but usually good cells that last the experiment. If you over-digest it, you think it's great because you have a large yield and the cells look good, but they'll beat for about a minute and then they start to fibrillate. So it's hard. The movie goes on, but that's maybe something for a different webinar. So you get your system ready, you turn on the microscope, you turn on the computer, the FSI, etc. Then you prep the microscope. We actually have a pacing insert that we connect everything to. That's the one. First, we connect the temperature sensor, so you can actually measure what the temperature in the bath is during the experiment, the inlet for the perfusion, the electrodes for the pacing, the outlet for the perfusion. Actually, in this experiment that we did this day, we didn't do perfusion. We just pipetted the ISO in, but you actually can. Then we put in the cells. The electrode set needs to stay in the incubator for a while, in the microscope for a while to warm up. And then we just put the cells in there, we put the electrode set in there, and then we're good. Next thing you do is you turn on the stimulator. We pace at 2 hertz. Mouse myocyte seems to like it. 6 volts, that's enough. And we're pacing, and we can start the measurement. And then off you go. We go to the software. What you just saw was a rapid scanning of an area in the dish, 3 by 3, and we can do that because we have a fast-moving objective. You set your trace recorders up to the appropriate parameter. You go back to the other window. You click on the overview to the cell that you'd like to see. You click on it to position it and rotate it. You check if your sarcomere signal is good. You see that on the moving cell on the left. It actually has a, and then you say start, and there you go. That is actually your experiment. That was the first measurement. You click on the next cell. Sometimes you have multiple good cells in the same field of view. You adjust it again. There's your second measurement. Let's rescale a little bit so we can actually see the data. People may say, you don't have a very strong contraction, and that's true. We only use 1 millimolar of calcium in the bath, and that's because we want to measure changes in calcium. If you use 1.8 millimolar calcium, you find it's hard to still increase your peak calcium. And if you pace at 2 hertz, 1 millimolar seems to be fine. I was going to say something, but I jumped away again. Normally, you would have 2 screens because it makes the workflow much easier. But if I do a screen recording with 2 screens, the resolution gets too poor. You see a stronger contraction correlates with a stronger calcium transient. And if you see the cells there that have a green box around them, those are the cells we already measured, so you won't measure them twice. Well, this will go on for a while. So let's jump to the last part of the recording. I think we have measured 27 cells now. We plan to do 25, so that means that we had 2 bad cells. We had to do some extras. So that we clicked on complete. So the first run is complete. It jumps back to the first cell, and you can start remeasuring it. What do we have to do now is actually load it with, not load it, we have to put the ISO into the bath. We did that. We waited a few minutes to incubate it to get a response, and you click automatic measurements. Now the computer is doing all the work. You see that it kind of refocuses and repositions it. We're not doing that, the computer is doing that. It finds the best focal plane for the sarcomere measurements. It rotates it a little bit, and then it does the measurement again. As you can see, it goes much faster now. So if you look at the cell that you see there, refocusing, repositioning, measurement. And at the end of the measurement, it goes boom to the next cell. This is all in real time. And you measure the next cell. And you see pretty much all the measurements are in the second run are good. This was actually a nice prep to be working with. It will keep doing this. Obviously, we have an effect. It will run through 25 cells again. I'm not going to show that. I'm not going to show all that to you. So let's move to the end. Oh, by the way, there's a reason why we didn't do fully automated cell detection in the first run. And because the plating density in the dish was too low. If the plating density is low, the computer just has to search much longer. Where if you have the 3 by 3 overview, you can just see where the good cells are. So it's the only way to make it go fast. So you have to be a bit pragmatic at times. So the experiment is finished. We save the data, and you see the overview. The first run, the second run, you see this clearly in effect. We collected backgrounds here and here. We messed up our data collection a little bit here. This was actually a human error. You can't blame the computer for that. And we actually have a mode in the iWizard that has a real-time overview, and the next slide has it annotated. This gives you an impression of how the experiment went. This the first black thing is actually measurement. Then you see the red shaded area, that's a pause where we find the next cell. Then we go to the next measurement, etc., etc. The long shaded area is where we actually incubated the cells with ISO. And then the real recording of the 25 cells only took 3 minutes because the computer moved very rapidly from cell to cell, so there wasn't a long pause in between. So once we've done that, we go to the data analysis. We load the cells, and you have to wait a little bit. It's actually analyzing the data as we speak. And as soon as it pops up, you can actually look at the data. You can load up to 20 files at the same time, but it seems to be a little bit silly for this demo. In blue is good data, red are rejected data. You can look at the different samples, at the transients. You have all the parameters associated with your calcium contractility transient. Here we isolate every single myocyte, but you can actually also analyze just the averages. Sometimes something gets rejected, and you can look at the reason why it was rejected. In this case, the signal to noise is too poor, but it is included in the average. So if you have poor, data with a lot of noise, you can average them and still do a good analysis. Now we need to annotate the data. The computer doesn't know yet what is ISO, what is baseline. So we actually create an experiment. We tell it what species we work with. We tell it the next grouping level, what we did, baseline versus ISO. Baseline. The next one is ISO, and this allows us to add this information to the data files that we just collected. So once we've created it, we say OK. Then you go to data annotation, which in this case is very easy because we only had one file. And 2 conditions. If you have a more complex experiment, of course, this will take a little bit longer, but not that much. So now the data are annotated, and we can export the data to a text file. We include the actual raw data, so we can show the average transients. I will go to the next part of our program. We import what we just collected. And what you see here is actually the average calcium transient and the average contractility transients from all the data. The shaded area is the confidence interval. We can also look at it normalized, and we'll look at the combination traits, calcium versus sarcomere length. But it gives you a very quick overview of what has changed in your experiment. I mean, it's not statistically significant or anything. You need to test for that. But as soon as you see that, you know what we're actually doing. You can explore your data. I know there is one outlier in this data set, and that's that one which has way too high baseline calcium. So what I would do is I will go back to my original data. For every cell, we store an image. And you look at the image to see if there's something odd, for example, a dead cell attached. If there's a reason to reject it, and so on. We have summarized data like a box and whisker plot, and it's very clear that contractility increases when you add ISO. That's not unexpected. It's a pretty standard experiment to prove that point. You can go over the data and see the actual values. And you can also look at the number. We can also present a table, and on the left you can select what you actually want to show and which numbers you want to look at. We don't intend to replace a graph path. This is just an easier and quicker way to have an overview of your data. All right. In one of our previous webinars, I had a hypothetical question. Can we do over 1,000 myocytes per day? Actually, if you do non-repeated measures and you don't measure calcium messes it up a little bit because you have to load it with ISO. If you do non-repeated measures, you have a decent quality of cells, you proper density and plating, you measure for 5 seconds. The answer is 18 seconds per cell. On average, we automated finding takes 30 seconds. If you have perfect plating, you can do it a little bit faster than that, but this is about it. So theoretically, you can do 200 cells per hour. There's always overhead, you have to change dishes, you have to add compounds, etc., so the number is reduced. But in practice, over the past 20 experimental days, we did 125 to 150 cells per hour. We measure for about 8 hours, so for mouse myocytes, we now really do more than 1,000 cells a day. This shows you another example of a data recording. This was done on large animals, so the contractions are much lower, but you still see a very clear ISO effect. It wasn't done with our high-speed microscope. We actually also support prior motorized stages. And although it may take you 1 or 2 seconds to get to the next cell, you can still do repeated measures on a lot of cells at the same time. These were the first published results with the system. Rio Juni at Amsterdam University Medical Center. He had a complex experiment. He wanted to co-culture endothelial cells with cardiomyocytes, with controls, and he wanted to test a bunch of compounds and combinations. So he had a pretty large matrix of things he wanted to test. The only way to achieve that was by using our system. These are some figures taken from a publication by the group from Imperial College, a group from Steve Marston and Peter Wright wrote it. Where you see in the top left, you see the drug effects of something that slows contraction, increases contraction, or a conjunctive that slows down contraction. And the C part of the graph I put in there because it nicely shows you can take multiple time points for each myocyte and still follow 10 cells. So they found 10 cells. I think they measured 10 or 15 seconds per cell, but by rapidly cycling through it in 16, 17 minutes, they could still get 10 time points for each cell. So not only can you measure the effect on a large number of cells, you can also get the time course. So the summary is we can greatly increase the yield of results per animal, which allows you to reduce the number of animals, although reality usually is you don't reduce the number of animals, you increase the experiments that you do. But it does allow new experimental strategies because you can test many more compounds or you can use the power of large data sets. And last but not least, I think using the analysis to get very rapid feedback on the progress of your project is very important to make sure that you don't discover at the end that you did something wrong from the beginning. We can make data acquisition easy. The hard part is still cell preparation, whether it's the cell isolation or to subsequently carefully treat your cells, load them very consistently, because all those things add to variation that you don't want to have. So I'm not saying the experiment is easy. I'm just saying that we made the data acquisition a bit easier. What is in the pipeline for us? By now, we actually have a multi-well plate support for our microscope, and the data Diederik showed have been collected with multi-well 24-well plates. We're almost finished with full climate control, where you control the temperature and the humidity and the CO2 percentage, which should allow you to do much longer experiments. And a movie that I wanted to show you because it's pretty cool, although I hope that your screen resolution is better than mine. In the movie shows the monolayer of cells, and the computer automatically finds areas that are contracting together concentrically, and then measures the contractility using a correlation algorithm. And this allows you to collect a lot of data from multi-well dishes with stem cell-derived cardiomyocytes, which we're pretty excited about. On that note, I want to thank you for listening to me, and I want to pass it on back to Sarah.
Thanks, Michael. So we're going to do a quick audience poll again, so you all know how this works. So this question is, which cell types do you use in your research? And please select all that apply. So we have adult cardiomyocytes, neonatal cardiomyocytes, ESC or iPSC cardiomyocytes, cardiac tissue, which is papillary, trabecular slices, etc., whole heart, engineered tissue, or none of the above. So there's a lot of options here, so I'm going to give you guys a couple more seconds to make your selections. And it looks like we have most answers, so thank you for participating again. And we are going to move on to our next speaker. So I'm very pleased to introduce Dr. Diederik Kuster. Diederik, the floor is yours whenever you're ready.
Thanks, Sarah. What I'm going to do is going to talk you through some real-life applications of the multi-cell system as we've been using it in our lab. Basically, we're going to do three small projects. The first one would be the traditional approach, and then we're talking about our first large-scale measurements, as Michael already introduced, and then finally some unpublished data of a project in progress in which we do compound screening. So first, I'm going to give you a very brief introduction on the disease. Otherwise, this will be not so informative. We research hypertrophic cardiomyopathy, which is an inherited cardiac disease, which is quite prevalent. And in the disease, you see both hypertrophy, so growth of the heart muscle, and diastolic dysfunction, so relaxation issues. And even though we know the cause, which are mutations in sarcomere proteins, the disease process is very poorly understood, or quite poorly understood. And what we did is we did a large-scale proteomic screening in patient cohorts, so on myocardium of patients with HCM. And what we found is, one of the things we found in this proteomic screen is that microtubule remodeling was one of the key processes that was disturbed. And we saw a large upregulation in the number of microtubules in these samples, and also one specific post-translational modification of microtubules, being detyrosinated tubulin. So this is a modification of tubulin, and it has previously been shown by, amongst other, the Prosser lab and also the Ward lab, that these detyrosinated forms of tubulin actually can bind to desmin and lead to increases in myofilament stiffness. And we see in these HCM samples a massive increase in this detyrosinated tubulin modification. So what we want to do is to see if this increase in detyrosinated tubulin actually contributes to the diastolic dysfunction that is seen in these patients. And to do this, we used this compound called partinolite, which is known to block tubulin detyrosination. So we incubated cardiomyocytes, which we isolated from our HCM mouse model, and we incubate them with this compound partinolite or with DMSO, which is the vehicle control, and then we measured contractility and calcium transients. And what you can see here on the left, you see the calcium reuptake kinetics, because what we're most interested in is relaxation parameters. And there's four groups. You see the wild types, either treated with vehicle control or with partinolite. And then you see the HCM model, same thing, so DMSO or PTL. And basically in the calcium reuptake kinetic, we see a very little difference. And we actually see slight increase in the time to baseline in the HCM mouse model, but partinolite did not have a big effect. But if you look at contractility data, and this is data from 4 wild types, about 200 cells in the DMSO and about 100 in the PTLs, and 6 HCM mice. And then you see that there's actually quite a sizable increase in the time to baseline if you compare just the wild type versus the HCM model, which we already know. And then treating these cells with PTL decreases the relaxation, and it does so quite almost back to wild type levels in the HCM mouse model. So this was a basic experiment in which you just treat a compound, and there's 4 groups. But this made our experiments much quicker because we got to measure a lot of myocytes instead of 10 to 15 per day. So therefore, but then again, so this gave us just in a few measuring days and these results, which are currently in press. So for the second part, I think we used a little bit more the capabilities of the machine, And this was actually just almost like a proof of concept study in which we want to use the system to see if we could detect differences between cardiomyocytes isolated from different regions of the heart. As you would know, it's the atria do something very different from the left ventricle, and the left ventricle does something quite different from the right ventricle, or it does the same thing but at very different differing pressures. So what we did is we isolated, as you would normally do, with the Langendorf digestion with Liberase or Langendorf system. We used Liberase to digest the hearts, and before we do the final step, we separate the heart in different regions, so atria, left ventricle, right ventricle, and the septum. And then so we do the last part and the calcium equilibration in separate in separate tubes, and then we plate them on different wells or different dishes. And then we measure the cells again at 2 Hertz, as also Michael alluded to. And then, well, basically what you can see if you look at these cells is these ventricular cells look quite different than the atrial, or the atrial cells look quite different from the ventricular cells, if you want. And then, but for the rest, the ventricular cells look quite similar to each other. And what we did is we took from these 5 animals 2,500 cells, and what we did is we did some inclusion criteria. And I think this is a key step here, in that you have to set these criteria preferably before you start. And what we did is we, for instance, needed or we are one of our criteria is more than 4 fitable contraction tensions per cell, to not base your data on 1 or 2 contractions and the rest artifacts. And also we required the fits of the different parameters to be of high level. And then we did this hierarchical clustering analysis that was already mentioned before from the Sickle paper to look at differences. And yeah, so obviously you can just take those couple of thousand or hundreds of myocytes in each group and then just look at, make some bar graphs. But I think these high number of samples require also some different types of analysis. And if you do a principal component analysis, you see on the left with all the data input, and then we use selected parameters for contraction, relaxation, and also contraction kinetics. Then you see, or you might appreciate that the purple dots are the atrial cells and all the other dots are ventricular myocytes. The atrial cells cluster together a little bit, while all the ventricular cells form another cluster. And even if you leave out the atrial cells, the different regions from the ventricular cardiomyocytes do not cluster in separate clusters. So they are much more alike than they are apart. And if you do if you take all the cells of the different regions together and look for averages, average transients, you might appreciate that the atrial cardiomyocytes have a much smaller contraction, and also that their kinetics is slower than the ventricular ones. And there might be small differences between the ventricular cardiomyocytes, but they are actually very subtle, and they don't explain why your right ventricle, for instance, contracts or has a much less force generating than the left ventricle. But it also allows you to do some other cool things. For instance, look at correlations between different parameters. And so this might be a little bit hard to read if you're looking at a small screen. But what we do, on top screen, you see different parameters such as your baseline sarcomere length or the percentage of shortening. And we looked at correlations between all these parameters. So if you look, for instance, in the top left, you see baseline versus baseline, but that is an obvious. But if you look at baseline versus percentage shortening, you see that there's the R squared is 0.007. So there's a very poor correlation between your sarcomere, your diastolic sarcomere length and the percentage shortening in your subsequent contraction. So there's actually quite a poor correlation between a lot of these parameters. But what was striking is that there was actually quite a strong correlation between contraction velocity and relaxation velocity, and contraction velocity and percentage shortening. So what we actually see is that the contraction velocity and so how quickly you contract is very predictive for how much you contract and also how quickly you then relax again. So this held up within all the cardiomyocytes of the different regions. So these type of analyses are now possible. And also something that was also useful for us for a later study is to be able to get very good estimates of variation in different parameters. So what you're seeing here is on the y-axis a sample size per group that's needed to, on the x-axis you have the relaxation velocity differences. So what you say if you have two groups, treated and untreated, if you want to study a 20% difference in relaxation velocity, so a 20% increase or decrease, doesn't matter, then you look at 20% on the x-axis, and then you have to go up, and then you see either the dashed line, which is a p-value of 0.01, or the dark line, which is a p-value of 0.05. So if you want a 20% increase in relaxation velocity, if you want to detect that, you need about 100 cardiomyocytes to reach p-value 0.05, because there's quite a large variation in relaxation velocity parameters, or relaxation velocity. Yeah. So this for us is very useful to go on to if you want to detect differences in different parameters. So, and then the final part, which is exciting because this is not published yet, and this is still ongoing as we speak today. I think there's an additional measurement. So what we want to do is we want to identify novel modulators of contraction or relaxation using an unbiased approach. And what we do is we use a compound library of 160 kinase inhibitors, and then we want to test the effect that each individual inhibitor has on adult mouse cardiomyocytes. And then our approach is that we test each compound initially in 2 animals, and then for each experimental day we aim for 30 cells per compound per animal. And then if we take positive hits from those 2, so each compound is tested is twice, and then so if they're positive in both and reach a threshold, then we screen them further. So to do this, we had to overcome some technical challenges. So we are testing kinase inhibitors. So to show an effect of an inhibitor, the kinase has to be on, so the signaling has to be on. And if you isolate cardiomyocytes, they are typically either in some kind of medium or in thyroid solution, and we usually measure in thyroid solution. So some of the signaling pathways will not be on. So this is one challenge to overcome. Also, the question is, how many control measurements do you include? So preferably a lot, but then again, every control measurement, you could also test a compound. So this way you will lose throughput. So how many control measures do you have to include? And also control for like rundown during a measurement day. We know that mouse cardiomyocytes are not so stable. So how do you control for that, and how much rundown is there actually? So our approach, as we do it now, is we use 2 24-well plates. We do have the multi-well system. They are laminin coated, and then we plate the cells in plating medium, which contains 5% serum. And then after plating, we exchange to tyrode. At least the plate we're measuring, the other one we exchange later. And then we measure at 37 degrees, try to keep the temperature as within 0.5 degrees of 37. Try, this is achievable. And then we measure for a max of 15 minutes per well. And usually if the cell isolation is good, this is achievable very easily to get 30 cells within 15 minutes. And then we randomize compounds and we use blinding not to interfere. So some of the technical challenges I mentioned earlier, so the activation of signaling cascades. So if you want to test an agonist, and this is also shown in the previous part, if you want to show the effect of ISO, this is a 15 nanomolar concentration of ISO. This is the easiest thing, right? This is our positive control always. If we want ISO, show the ISO effect, so we now have fractional shortening. Well without ISO, well with ISO, you see a huge effect. But if we now use an agonist for the same receptor, so if you now use a beta blocker, so propranolol, we actually see no effect if we do this in tyrode. This is also not so surprising because there's no adrenaline or noradrenaline in your, or ISO in your tyrode. So if you use, so this is a challenge. So one of the solutions we came up with is use some serum. So if you want to test one receptor, you can also add an ISO, of course, but because we want to test many different receptors, we want something that turns on a lot of different things. And then we tested serum as a collection of a lot of growth factors and other things. And then we show that if you give some serum, you can still see the ISO response. So this ISO plus serum is control plus serum. But if you now add propranolol in the serum conditions, you see a difference from control. We tested for different concentrations of serum, 2%. If you go higher, it still shows an effect, but the cells become less stable. And 2% they can keep going for the rest of the day. So this is why we now measure in our condition with 2% serum. Yeah, so now the second question is, okay, so how many controls do you include? And we add quite a lot of controls. So what you see here is data from one measuring day. This is from, I think, 2 or 3 weeks ago, in which we measured 16 compounds on a day, and we measured 8 controls. So what we do is we have compound 1, and we are now looking at fractional shortening. So these are about 30 cells. Then we have a control. We have compound 2, compound 3, another control, compound 4. So we add, each compound is neighbored by a control. Because this is a picture we see almost all the time, in which there's a slightly higher contraction, and also kinetics are slightly higher in the beginning of the day. Once they're just, these cells are isolated maybe an hour and a half ago, they are have a slightly higher contractility than cells that are in the medium for longer or in tyrode for longer. So that's why. So let's say compound 2. Now we're pretty sure that this leads to an increase in contraction because it's much higher than the control value, or considerably higher than the control value. And this is also why we randomize our compounds, so that we don't always measure compound 2 at the beginning of the day. But if you would use absolute numbers, I think you will miss a lot of this information. So this is why we choose to do this, and then we normalize every value to its neighboring control. And we do this for all parameters. So here we measured 1,056 cells, of which we can include 783 cells in the analysis. So we lost some cells due to bad fits or artifacts in the contractions. So, and this is then the data you can get out of this. So what we show here in the normalized contraction, normalized fractional shortening. So this is each compound divided by its neighboring control, and the average taken of these two measurements. It took us 21 measuring days to get this data. So, and then you see all the 160 compounds. They produce less contraction if we treat them with an inhibitor, or they produce more contraction if we inhibit a kinase. And as an example, this is serum glucocorticoid kinase 1, which is a known activator of contraction. If we block it, we see reduced contraction. So there's a number of these compounds, but we know that should increase contraction. If we block it, they decrease contraction, and vice versa. And so we get this data from not only for fractional shortening, but also kinetics of contraction, kinetics of relaxation. And so this is a way to get perform compound screenings. So to conclude, we know that large-scale measurements are feasible, and they're actually easy to do, and I think more easy than with the previous setups. In our hands, the 24-well format has considerable advantages. And the handling of dishes is easier, but also temperature control is easier. But you can, of course, also do this in 35 millimeter dishes. We did the atrial versus ventricular cardiomyocytes all in 35 millimeter dishes. And so we are now doing the first large-scale compound screen for contraction relaxation. But what's still important, even though you can automate everything, is experimental artifacts are always there, whatever experiment you do. So be aware of those and curate your data well. So make sure that you actually see what you get, and make sure that you don't fall in the trap of believing your data because it looks good. So that's why building good controls and set inclusion criteria beforehand. So for my part, I want to thank a number of people. Valentine, Jans, who did a lot of the isolations, and Mike, and also a lot of measurements with Mike, which is a talented PhD student, and Jolanda van Velde is my supervisor and chair of the department. And as well as some people of Ion Optics that are listed here, such as Patrick and Emmy and the rest.
Awesome. Thank you so much for that wonderful presentation, Diederik. We're going to run one last audience poll before we get to our live Q&A session. So this poll is, do you currently acquire calcium and/or contractility data from isolated myocytes in your research? Yes or no? So we'll wait a couple seconds for everyone to answer that. Great. Thank you for your participation, everybody. And as those last couple answers roll in, we're just going to introduce our speakers back. I just want to make sure that you guys are here, Diederik and Michael.
I'm here.
Yeah.
Great. Okay. So we're going to move on. So our first question here is for Michael. Michael, this question is, what other indicator dyes have you tested?
We have mostly tested calcium dyes, fluo, rhodamine, Fura2, Fura4. We've also tested fluovolt. So we have done a range of things. There's not that much else that we're interested in at the moment, but it is a general fluorescence system. So pretty much any dye you can think of that you can measure in live cells with a photomultiplier too would be applicable.
Great. And this next question is also for you, Michael. Have you measured action potentials in iPSC cardiomyocytes?
We haven't, although we're very tempted to try it because we saw some data from another user that collected them in iPSCs with our system, and the data just gorgeous. It actually works better than adult cardiomyocytes. The signal is a bit better and doesn't suffer as much from cardiac toxicity. So we're very inclined to try the cells. If you do that, we'll, of course, share
Great.
the results.
Yeah, that's awesome. I'm sure everyone will be excited to see those results. Diederik, this next question is for you. This person has asked, how reproducible are the effects you see on the screen as you only measure the compounds twice?
Yeah, so so what we do is we measure each compound in 2 different animals, and then in between 30 and 50 cells per animal. So that data is relatively reproducible. Sometimes we see a little bit differing effects, and therefore all the positive hits that at least are positive in one of the 2 animals, we repeat a 3rd or a 4th time just to confirm. But all in all, it's pretty reproducible.
Okay, great. And this next question, Michael, is for you. What criteria does your software use to determine healthy myocytes?
We don't so much determine health, but you can select on, because that's kind of subjective. We do check for decent sarcomere striation. We can check for length and size. We can reject the ones that are too small or too big. And we can set a minimum sarcomere length. But there's more acceptance rejection criteria. It doesn't exactly tell you about health.
Okay, and so it does that automatically?
It does that automatically. And of course, you can still select pulse lock based on the data, like whether there's artifacts in the cells. You could say, well, the cell has to be fast and over. You have to
Cool.
take an open view as a researcher.
Really cool. Okay, this next question is for you, Diederik. This person has asked, you use kinase inhibitors in your screening, but most of the compounds increase contraction. Is that what you expected?
Well, it was not what I expected, but once I saw the data, it made sense, or at least it could make sense, because one of the issues we have is that it's easier because the cells are contracting, but they contract not very strongly if we don't give them anything. So we give them serum, then they contract a little bit stronger, but they still do not contract very strongly. So therefore, I think activating it is a little bit more easy than inhibiting fractional shortening. So in that respect, I think that's part of the answer. But apparently also a lot of kinases, if you inhibit it, that will activate contraction. So apparently there's a large set of these kinases in your cell, which is interesting. And we're following up on this.
Awesome. A future step then.
Yes.
Okay, we have another question here. I think, Michael, this one is for you. This question is, could you please elaborate a bit on how the solution exchange is done in the system? For example, the time course, the heating, etc.
The temperature is extremely important for your kinetics. So whatever we add has to be at the same temperature as the cell, so it has to be at 37 degrees. You can get beautiful dose responses if you perfuse in the solution, but it does have the downside that it takes a long time. We are finding that in a lot of cases, by replacing part of the solutions with solution that has the compound you want to test in it, also works, and it's actually a faster way to spread it around. So the isoproterenol experiment that I showed, we actually pipetted it in, and in the 24-well dishes, if I'm correct, we pipette 4 wells at a time, and then we measure them. Is that correct, Diederik? I think Diederik is muted now.
No. I was just actually reading the question, so I was not. Sorry, I was not listening to you, Michael. Can you repeat it?
Are we pipetting compounds into 4 wells at a time and then measure them?
So we pipette them in per well,
Okay,
and then measure it.
So open the lid, pipette it in, close it, measure it.
Yeah, so we just, exactly. But we take out, we don't add small volume. So we take out a little bit, 250 microliters, and then pipette that back in so that we don't get problems with mixing or slow diffusion of compounds. That works well for, yeah.
So it's clear who's actually doing the experiments here.
Great. So we have hit the noon mark, but there are a ton of questions, so we're going to keep going. But I did want to remind everybody that if you can't stay with us, that's okay. We are going to be posting a recording of this webinar. So if you wanted to come back later and watch the rest of the Q&A when you have more free time, you can absolutely do that, and you'll receive an email when it's ready. Before you go, though, we do want to bring your attention to this survey that just popped up on your screen. We'd love to get your feedback on the webinar, and if you have any additional questions, you can submit them here on the survey or using the Ask a Question panel, whichever you prefer, and we'll make sure that those questions get back to our speakers. But like I said, we are going to continue to answer some questions, so if you can stick around, that's great. I'm sure these questions will be riveting and the answers will be really informative. So I'm going to continue on with the Q&A, but please do take a look at that survey before you head out. Okay, so our next question is here. I'm not sure who it's for, but the question is, you measured more than 1,000 cells, but you only included 700 cells. What was your exclusion criteria? And I think based on the time that it came in, it's for Diederik.
Yeah, I think so. Yeah, so this is also based on fits of the data. So if the data could be fitted well. Yeah, sometimes you have an artifact if there's too much shortening, or if there's something, the sarcomere pattern is not as clean as you thought it was when you started measuring, that you get an artifact in your traces. Yeah, these are the exclusion criteria. So basically, inability to get good fits and therefore good data.
Cool. Thank you for that answer. It's really great. We have a couple more questions here that I'm going to just keep going with. So, Michael, this question is for you. Which software are you doing the analysis in?
We created, Cytocipher created the Cytosolver software because we knew that we would hit a bottleneck if we didn't do that. And that has been evolving. And as soon as we were able to collect many more data, we realized we needed to make the next step easier as well. So we used something called RShiny that allows you to really quite easily build dashboards to connect to our database and create graphs from the data you just analyzed and do it per project. So we're trying to go to a really project-based approach. So yes, it's our software. You can get access to it.
Okay, makes sense. There's another question here. Is this high throughput system applicable to isolated skeletal muscle myocytes?
Can I answer that?
Yeah, go
ahead. We have done it with FDB muscle. FDB muscles they're about 3 times the size of cardiomyocytes, and if you plate them and you paste them, you can actually you can measure them the same way. It's a little bit difficult because they're really fast, so we actually had to go to at least 500 frames per second to capture the contraction. And they tend to jump if you twitch them. So we actually had to pour some collagen over the cells to keep them in place. We spent some time on it, but it's absolutely possible.
Okay, great. So lots of applications there for people who do skeletal muscle research too. This question is for Michael, I believe. This question is, which isoproterenol concentration did you use?
I used 15 nanomolar.
Okay, great.
We usually use 15, but we wanted to be really sure about the effects, so we used 50 nanomolar.
Okay. Awesome. And we have another question here. How easy is it to detect arrhythmic events? And can calcium dyes interfere with the detection of these arrhythmic events?
I can take it, I guess.
Okay.
Calcium dyes always confound your experiment to some extent because you add a calcium buff to your solutions. You should also always be aware of it. So I can imagine they can also dampen out some sparks, but I'm no specialist on that in that area. Our software does have analysis algorithms that can detect after contractions and the likes. So we have some arrhythmia detection capability.
Okay, cool. And the next question here, I believe, is for Diederik. When measuring sarcomere length data, how long does it take to find the right direction / focus / location?
So if you do automatic self-finding, it does so automatically, of course. So that's very easy. If you do it by hand, it's also very quick. So there's basically you have to click on the cell, and then you rotate it using either a mouse or controls on your keyboard, and then you can, if you want to measure the different spots, you just click somewhere else with your mouse. So this is much quicker than the old method in which you had to rotate the camera and adjust the window. So it's very quick. It takes a matter of seconds.
Okay, cool. That's good to know. And I think this will be our last question. Just want to be respectful of everyone's time. So this question is, why did you select TTB of 70%? And did the other TTB values have the same significance? And also, as a second part to that question, why not use tau from mono exponential fit or bioexponential data?
Yeah, so really good question. So in our paper, we
Oh, Diederik, we're losing your sound a bit.
Do you want me to take over?
Sure, yeah.
If Diederik comes back, he can correct me. Yeah.
Okay.
We had to pick one, and it seemed that because you can't show everything, and 70% seems to be the most consistent value. Tau is great, but you need to take great care about how exactly you fit it, because it depends on your data shape. So I find it harder to automate and to get really good data. Plus, when you have contractility data, the sarcomere shortening, it's really not a single exponential return. So personally, I always had trouble using tau for that. And I'd rather use time to percentage shortening. In the end, they give very much the same data. They give the same answers qualitatively.
Okay. Diederik, are you back?
I'm not sure.
Yes, you are.
If you can hear me.
Yes, we can. Great.
Okay. Yeah. No, I agree. So in the paper, if we publish it, we'll publish as much data as we can. But yeah, I agree with Michael. They do show the same things, and tau is a little bit more finicky, I guess. Times the baseline, okay, you always get correctly.
Great. Awesome. Well, yeah, I just want to take the last couple seconds of the webinar to thank Michael and Diederik again for your fantastic insights today, both in your presentations as well as the Q&A session. And I also wanted to thank everyone who's still here for joining the webinar. Today, we really appreciate you being here live and all of the wonderful questions that you submitted. Like I mentioned, the slides and a recording of today's webinar will be posted and available pretty soon, so look out for an email from us that gives you access to the video recording in the near future. And as I mentioned before, we do have the survey, so before you go, I'm just going to make sure that everyone sees that survey. Please give your feedback. We would love to hear from you. And if you do have any last minute questions, you can submit them through the Ask a Question panel before you go. In closing, thank you again to everyone for taking a part in this webinar. We want to thank our sponsor, Ion Optics, one more time for producing this wonderful event. It's not possible without our sponsors, so send lots of love their way. And we look forward to having everyone with us again soon. Have a great day, everyone.